Hello everyone, my name is Michael Downy, Library and Information Services Coordinator at the AWRI, and it's my pleasure to welcome you to today's webinar, the fourth for the 2015-16 program. Now, a slight change for today. Barbara Hall is unavailable, but we're very lucky to have Suzanne McKay, from our Senior Research Officer with SARDI, who has worked on the project with Barbara, to fill in. Now, the topic remains the same. The topic is understanding fungicide resistance in Australian vineyards. Before I go to Suzanne to tell us more about today's topic, a couple of quick notes on what you as the audience should expect from today. The format is a short presentation followed by a Q&A. The session is being recorded and will be available to view via the AWRI website. And finally, for those that are new to webinars, it's important to remember that webinars are interactive. There are a number of ways to get involved. To ask a question, please type into the question section of your control panel in the bottom right-hand corner of your screen. For those with access to a microphone, there is also the option of speaking directly with our presenter by clicking the raise hand button. This is located in the top left section of your control panel. If you'd prefer to ask a question via email, please do so at infoservices@awri.com.au. Please feel free to send through your questions at any time, and once we get to the Q&A, I'll get as many questions through as possible. Finally, if you'd like to get involved through Twitter, please use the Twitter handle at the AWRI, at the _AWRI. Okay, that's enough from me for now. I'm now going to hand over to Suzanne to tell us more about today's topic.
Okay, thank you. Welcome everybody. As Michael said, I'm stepping in as Barbara is unavailable. So let's get started. I thought I'd start with some general background about resistance that many of you are probably very well aware of. However, in terms of practical resistance, we deem that to be a loss of acceptable control when the chemical is used at label rates. Not above rates, not below rates, but at label rate and label frequency. It's important to notice that resistance is actually caused by selection of resistance individuals in that population. The fungicide doesn't cause the fungus to become resistant. We'll talk about fungus today, if that's okay, because this project is only about fungal diseases. So the fungicide causes selection of these resistant individuals that already exist at low levels in the population. Now, these resistant individuals need to survive, and then they need to reproduce. Okay, so if we just look at this slide for a moment, if we're just showing here on the left-hand side the initial population, the yellow dots represent the sensitive individuals in the population, and the red dot represents a naturally occurring resistant individual in the population. So what this means is if this individual already has a mutation, the fungal cells in the population have many mutations, and some of these they just sit around, do nothing, don't affect the population at all until they come in contact with a particular chemical. The mutation occurs at the target site in the cell, so the target site where the fungicide actually interacts with the cell. If a mutation occurs at that target site, then the fungicide is unable to bind to the fungus, and therefore it can be resistant. So that's really important point to note, that the resistant individuals already exist at low levels within all the populations, whether they've been exposed to the chemical or not. Okay, so then we apply the chemical, we apply the fungicide. That fungicide kills most of the sensitive population, some survive, but it doesn't kill the resistant ones because the fungicide can't bind, the fungus isn't killed, and therefore it survives. So then if that individual is fit enough and the mutation doesn't cause any adverse effect on the cell, it then reproduces and builds up in the population, builds up in numbers. The chemical is then applied again Again, the chemical knocks off a lot of the sensitive individuals. However, the resistant individuals still survive. They then reproduce, and eventually the resistant population is where we have more or an abundance of the resistant individuals in that population, and this is what we may see as resistance in the field. But let's point at this situation here that we have in the centre. This is what we don't really understand. We have a mixture of these sensitive and these resistant individuals, and we don't really understand how does that translate in the field. We've got some that are resistant, some that are sensitive. What's the proportion of sensitive and resistant that we need to be able to see resistance in the field? We don't understand that really. Okay, just briefly looking at some types of resistance without going into too much technical detail. First, we have what the two extremes are, where we have a sudden loss of field control or a gradual loss. So what we see in the top graph is the blue. If we look at the blue bars, this is a population of the fungus in the field that has never been exposed to a particular fungicide. So what that means is we've got a low concentration of the fungicide is needed to control that population. Let's apply the fungicide at its label rates for 5 years, and what we see is a slight shift where we need slightly higher concentrations to control the population or control the disease. 10 years later, we are seeing we need high concentrations of the fungicide in order to control the disease. So here we see like what appears to be a sudden shift. But what's happening is, if I just go back a slide, what's happening is you're still getting this gradual increase in the population of resistant individuals. But what you see in the field is this sudden change of control to no control. And this sudden loss of field control happens with fungicides that are single site actives, and where the resistance is controlled by a major or single gene in that fungus. Not many genes, but single genes. And this is examples of the QOIs and metalaxyl. But what we see in a different situation where we're getting gradual loss of control. This is where we've got several smaller mutations perhaps, or interacting genes, or multi-genes, many genes with mutations that make the fungicide unable to bind, not just on one target site. And this is an example of, DMIs are an example of this. They are still considered a single site active, but they act in a different way than, let's say, the QOIs. So what we see in the field is this gradual loss of control. So we have, the baseline where this fungus has not been exposed to the fungicide. Then we have a shift 5 years and a shift 10 years. Now the 5 and 10 years, this is just experimental representation. It could happen over a year, it could happen over 20 years. So please don't take the 5 and 10 years as necessarily the actual time frame for every fungicide. So the gradual loss of control, small losses of efficacy in the field, the disease is not being controlled slowly, versus what you see as a major loss. So they're different types of resistance. Okay, and so the timescale for resistance development, it depends on many things. Firstly, the mode of action of the fungicide, as I've just said about single site actives, exactly where the mutation occurs in the actual fungus. So I said the genetics of the fungus, the target site, the fungus life cycle, that's really important. It's how quickly can the fungus regenerate, how well does it survive, those sort of things also affect the development of resistance. And the fungicide applications, the number and the timing. So if fungicides are applied more frequently, then you are exposing that resistant population to that fungicide, so you're going to get a faster build-up of resistance than if you apply fungicides less frequently. And again, timing is also critical here, because if you expose a population that's, let's say, powdery mildew at Christmas time, and where generally that's when the big outbreaks, people see it and go, oh my gosh, we've got really bad powdery. If you spray that fungicide then you are exposing this naturally occurring resistant number of resistant individuals in the population. You're exposing many more of them to the fungicide, compared to if you sprayed very early in the season with that same fungicide. So timing is really critical. And Trevor Wicks, who I'm sure many of you know, he's also very keen that we push that not every field failure or every disease control failure is due to fungicide resistance. Because of these other factors, timing is a very major factor and a number of applications. So not everything we see is resistance. But the key point here is to note that it is very difficult to predict the development of resistance. So before this project started, just a brief snapshot of the fungicide groups where resistance had been detected. So for the benzimidazoles, group 1, iprodione in group 2, and aminopyrimidines in group 12, so there had been resistance found in Australian vineyards to these fungicide groups for Botrytis, metalaxyl for Downy Mildew, and there had been reports of QOIs and DMIs to powdery mildew. However, the QOI resistance story is interesting because that's only been based on genetic results, not on actual field tests, not on actual tests in laboratory, but only genetic results. And genetic results can only give us so much information. So partly the reason this project started is, yes, okay, we've got these, there is some genetic basis for resistance that we've found in Australia, but do we really know what the incidence and severity of fungicide resistance is in Australian vineyards? So I'm currently working on the powdery mildew aspect of this project. It's a 3-year project. I'm due to finish June next year, so it's in its sort of final stages. Barbara Hall is the project leader, but we have a lot of collaborators. The Botrytis has been worked on by our colleagues at Curtin in Western Australia, and Downy Mildew by Sandra Savokia in Charles Sturt, New South Wales. So I'd just like to show you that we have a lot of industry collaborators and an industry reference group who have been invaluable in giving us input into the directions for this project. So it's not just us here at SARDI, it's a big group of people, and of course AWRI as well. Okay, look, I'd just like to show you briefly our approach to, so firstly what we do is samples were collected in first season was 2013-14 and last season. So we've got samples from as many areas as we could around Australia, and many of you may well have contributed samples to the project, so thank you. In the case of powdery and downy, the difficulty, I'm going to whinge about how hard my project is here, is that powdery and downy are obligate biotrophs, and that means they only grow on live tissue. So what has to happen is we had to get samples in really good quality, and where the powdery was still growing and still active, then I have to grow on live tissue, and I've got to do that every 2 weeks for the whole year, and live grapevine tissue. So that is one reason I think that the powdery and downy story is more difficult to unravel and has not been worked out, although powdery has been worked on for how many years now. It's a difficult disease to work with. It grows really well in the field, as I'm sure all of you can attest to. It grows well in the laboratory too, but it's a lot of work to keep the thing growing. We can't store it, we can't put it away and work with it later. So having said that, Botrytis is a different kettle of fish. It's much easier. We can grow it on agar plates. It can be stored. You can churn through a lot more samples, do a lot more testing very quickly and a lot more easily. So I guess that's probably why there's more information about Botrytis out there in terms of resistance. Much easier to work with. Okay, so we've got our samples. What happens then? We do a laboratory test for sensitivity, and again with powdery and downy that has to be done on live tissue, and whereas Botrytis can be done on Petri plates or in microarray plates and done very quickly. Then all those samples, each sample that we get in, we send to our colleagues in Western Australia, and they basically extract the DNA and they're trying to detect the mutation. So we're calling it the resistance gene, for example. It's maybe many genes, but in the case of QOIs it's one gene, and in the case of DMIs we're looking at one major gene. So they extract the DNA. Can we detect the mutation in that particular sample? And then from looking at laboratory tests and the detection of mutations, as well as field data that we ask from you when we get samples, to understand, has there been field failure? Has this fungicide, particularly let's say QOIs, been applied for how many years? So we're getting a picture of what we find in the lab, what we find in terms of genetics and what's been applied in the field. Yeah, so that's what's very important when we do ask for samples, that we do harass you really for getting information about your spray program, because it all adds to the picture. Then we can make a determination: is it sensitive, is it resistant, is it somewhere in between? And here, can I just point out, there is actually no, for powdery for example, no actual test. It would be lovely. You send the samples in, we run it through, bang, you can tell if it's resistant or not. That doesn't exist. It's, as I said, it's a combination of things to determine resistance, and it's not as simple as just one test, and I guess that's where we're hoping to head towards. Okay, so in this project we've looked at, if we just look at this table here, the fungicide groups with the red tick are what we're looking at in this project. Okay, so 4 groups within Botrytis, 2 in Downy Mildew, and the QOIs and DMIs. Now on this note, I can maybe just say that some people have said to me, well, why are you looking at the QOIs? We know there's resistance out there. And as I said earlier though, is that yes, we think there's resistance. Some of this information has only been based on genetic tests. Some there probably is, but we actually really don't know how widespread it is, and based on what evidence, I guess. Okay, so just briefly, sample collection, just to show you where we've collected samples. The blue dots in the first season, yellow dots last season, and to point out that in the first season, a lot of these samples were not suitable for testing. They arrived, they were picked quite late in the season, grapes were all mushy by the time they got to me, and because powdery has to survive, I couldn't grow it, so we couldn't do a lot of testing. So the second season we got much better, and we liaised better with industry, and we got some really good samples in, so we still might be looking at some collection this year. It depends, because I've just started testing this year's samples now, so that's a 6-month lag between getting samples and testing, and so I'm not sure how that will go next year, but we still may be asking for samples, particularly if you really think you have resistance issues. So I guess if you see a call going out for samples, then yeah, that would be great, but we're not sure exactly yet. We'll see how we go. Okay, so just on to some broadly the results. I'm not going to go into lots of detail because we're still trying to understand what's going on. However, looking at powdery, which is what I'm working on, this is just a map showing the distribution of samples with reduced sensitivity to Cabrio. Now I'm going to point out here, I'm not saying resistance. I think that's a big call to say, bang, you've got resistance. We, I guess, are scientists and often can be a bit conservative and don't necessarily want to go out and say, yep, you've got resistance. I think there probably is some actual resistance there, but we are calling it reduced sensitivity at this stage. So the red dots on the picture of Australia, or the lower half of Australia, show where I have found decreased sensitivity in the lab, plus that particular sample had 100% of it had the resistance gene. So these samples are likely, I think, to be resistant, but you know, that's again a big call, so I think we're saying decreased sensitivity. The yellow dots are where we have decreased sensitivity. However, this is a different part of the picture. We have a mixture. That sample has a mixture of resistance and non-resistance genes. So this is something we're just trying to get a bit more of a handle on. There's research in the literature that is saying that yes, we're finding mixtures of these genes. So what does that mean? If that one cell has 50% sensitive copies of the gene and 50% resistant copies of the gene within that cell, is that going to be resistant or not? What does that mean? And these are questions that we don't know the answer to, and I think are going to be important to understand in the future for be able to control disease for you guys in the field. Okay, if we're looking at existing, this is a slide from a colleague in Cornell in the USA, Wayne Wilcox. This area here had existing QOI resistance. So this is just to show that, okay, we have 98% of the clusters were infected with no treatment. Wayne applied pyraclostrobin, Cabrio in our country, or trade name Cabrio, at increasing rates, and basically there was no increase in control. So 7 sprays at 2 weeks intervals, absolutely nothing achieved by increasing the dose. And you know, of course, we don't recommend increasing the dose, but I guess there's reality in knowing what people do out there that can happen. But there's absolutely no control achieved. So this is showing that once you've got QI resistance, it's there. Doesn't matter what you do, it's there, and it's not going to change by chucking more fungicide at it. Yeah. This is also this is one of our experimental sites here in South Australia, where we know we had genetic QI resistance. It's a site where QIs were sprayed extensively as part of research, so we're fairly sure that there's quite a lot of resistance there. So what I'd like to show here is that, first of all, so last year I sprayed just 4 sprays, 2-weekly intervals from flowering, and the unsprayed had very high incidence, so the dark blue shows the number of bunches. Almost 100% of bunches had powdery and very high severity, so pretty bad. Sprayed with Cabrio, pretty much the same, no difference. So Cabrio here is ineffective. And what we can take, the message from this, is that QI resistance was there, it's been there for a long time, and it's still there. It takes, the literature says that QI resistance takes perhaps maybe 10 years to reverse. We don't really know, I think, but this does show that that Cabrio resistance was, or QI resistance was still there, loud and strong. Spraying with Myclos, what that did was still had a lot of bunches with powdery, but a much lower severity. So here we could say, okay, well, Myclos did control those Cabrio-resistant isolates really well. Topaz, even better, so much less decrease in incidence and decrease in severity. So this just gives an indication that changing that mode of action group, how control can be achieved of one group that's got resistance to one mode of action, and change to a different mode of action, and you're getting control. And also to take from this, although this is not a repeated experiment, this is just I was really trying to induce resistance to use in my tests, actually. That was the purpose behind this particular experiment. It wasn't an experiment to demonstrate this, it just showed interesting data. The difference between Topaz and Myclos, so not all DMIs are the same. Okay, looking at the distribution with samples with a DMI resistance gene. Now I'm saying only the gene here because in the lab I think we're considering all samples to be sensitive. However, many of them had the presence of the resistance gene. So this is a different picture than the QOIs, because generally there's quite a good relationship between the presence of the resistance gene and the decrease in sensitivity with the QOIs. DMIs are different. The resistance gene is like we call it maybe a precursor gene. It's the beginning of the resistance chain of events, I suppose. So if it's there, it's a warning sign to say, look, resistance could be developing, or it's a potential to develop. Now this story, DMIs, are very interesting, and there's still more to unravel that story. Okay, and as I said about all DMIs are not the same, this is also data from Wayne Wilcox in USA, just to show that all the DMIs aren't the same, basically, the different levels of control achieved with different fungicides. A couple of these, difenconazole and flutrifol, are not registered in Australia on grapevines, so this is just experimental data. So difenconazole, we're getting 75% or 70% disease control on leaves, and quite high percent control on clusters. Tebuconazole, much lower control. Flutrifol, still not very good control, whereas we've got tetraconazole at 2 different rates, and it was showing that there's actually quite a marked difference in the rate that is able to control the disease. However, please do not take this as an advice to apply more than the recommended rate. If you start applying this type of rate, then you will develop resistance much quicker, because you're exposing that fungicide, those resistant individuals, to much higher dose of the fungicide. You'll get resistance. Well, resistance is likely to develop, I should say. But it's just interesting experimentally to show that they are not all the same. So yeah, rotating even within the DMI, just because you're applying one DMI, you can be rotating within those DMIs to get better control. Okay, just moving on to Botrytis, so just a summary of the results for Botrytis. So we had, if we're looking at the 4 fungicide groups here that we tested, and this is colleagues Lincoln Harper and Fran Lopez in Western Australia, this is their work, not mine. So we had total number of samples, 7 had reduced sensitivity, 7 Oh, Barbara's changed the slide on me before so I think it's 7%. I think it's 7% of this sample had decreased sensitivity to this mode of action, 17%, 2% to Roverall, only quite low, but still a couple of samples, and 9. I'm sorry, actually, Barbara's changed the slide on me since I last saw it, so I actually just realised that I'm not 100% sure. Let's look at this one here, I think. Maybe that's the number of samples, and this is the percentage. So apologies for that. She caught me out there. Okay, the bottom line is that, okay, we've got 7% of samples, only 7% had reduced sensitivity to fenhexamide. Whereas we've got over, roughly a third of all the samples that we collected with reduced sensitivity to those other fungicides. Now, in the red, boscalid and fenhexamide, these are the first reports of reduced sensitivity to these fungicides, these fungicide groups. Mode of action, perhaps she's got the group here. That's what it is, I think. Yes, all right, that's what it is, the fungicide mode of action group. Yeah, okay. Right, so just moving on to the next slide, if we look at what this is showing is that the number of samples with reduced sensitivity to one or more modes of action. So 21% had reduced sensitivity to only one mode of action, 11% reduced sensitivity to 2, and so on. Now only 3%, this was one site each in SA and one site in Victoria, had reduced sensitivity to 4 modes of action. So this data in the red is also new, that although it's only fairly small numbers, it's still, I think, concerning that we have got some sites that are showing reduced sensitivity to 3 or 4 modes of action in Botrytis. I guess on the good positive side is that 54% of all the samples were actually sensitive. But I think it's a warning sign. Okay, just lastly, Downy Mildew. We had 61 samples that were received. 12 out of the 18 have been tested in the lab. I think there's more testing since this slide was done. And what we've found, what's new, Western Australia and Tasmania now have metalaxyl resistant populations. The mandipropamide, we're not sure yet, that possibly one resistant isolate, and genetic testing really is still very early days, so we're still not sure yet, but at this stage they're all negative for CAA and QI resistance genes. Okay, I'd like to finish off with just some of the other work that we're doing is the population studies, we call it. I guess what we want to know, what's the ideal number of samples to collect to detect resistance? How does resistance spread in your vineyard? How many samples do we need to collect? From where? That type of question. And how we're doing that is we're taking 100 swabs are taken from within a block. So I'll go out there with that swab, rub it in the powdery mildew. I'm not growing the powdery mildew, it's only used for DNA extraction, so we're not testing it in the lab for sensitivity. Then AWRI here, our colleagues are performing next generation high throughput sequencing, and what that does is determine the frequency of the mutant resistant genes for each swab. So in that swab we have a whole, if you swab the powdery mildew, we have a population of powdery mildew on that swab. And what we want to know is how much of that population on that swab contains the resistance gene. So I'll just show you, this is just an example of some of the results. So this is only 64 swabs because I can't fit the 100 swabs on the slide. What we have here, this arrow here, shows this sample, this is one swab, shows less than, or let's say about 15% of that population on that swab contain the resistance gene, 10%, 15%. This one, 50% of that swab contains resistance gene. This one, 100% of that swab, every cell in that swab had the resistance gene. And this is showing of insufficient samples for analyses. So the interesting thing out of here, I think this is, to me, where the project should be heading. This is the really exciting and really powerful methods, I think, to help us determine what does this mean. If we find this in your vineyard, what does this mean? Does this mean that you probably don't have resistance, or you're likely to get resistance? We don't know, and this technology and these type of studies will help us try and understand that. I think it will also, what we're hoping is to understand how many samples we need to collect. If I can just say, if we collected samples from here here, here, and let's say here, what would that show? It showed that, okay, from those 5 samples, we'd have a low level of resistance genes in your vineyard, and we might tell you, okay, that doesn't mean, yeah, look, you know, you're okay. But if we collected from this one, this one, this one, and this one, the results would come back, you have a high level of resistance genes in your vineyard. So what we need to know is how many samples are going to give us an accurate picture of what's happening in your vineyard. And also looking at here, if we're collecting samples, how much sample do we need? We don't, yeah, these are insufficient samples. So if we go out in your vineyard and the sample's not collected properly, we don't get enough sample, we can't analyse it. So those are some of the questions that this technology will help us answer. And I really think this is the way to go. And we are putting in money for, we're going to put in a bid for a further project. And I think this is going to be the area we're going to focus on. So in summary, I think, can we say just be alert and probably slightly alarmed. I think it's fair to say that we've found what is likely to be resistant populations, and I think it's always following the resistance management guidelines, which of course the AWRI guidebook, dog book is the Bible. Okay, so, and I'm sure most of you are highly aware of this, that rotate, use the label rates, limit your number of applications as per the guide in the dog book. Educate yourself. The more you understand about the modes of action and the resistance groups, then the more it will make sense as to how many you should apply, when and why. And keyly, do not use a single site mode of action fungicide to control well-established infections. So let's say at Christmas time, throwing on a single site active like QOI is probably not the way to go. So Paul Ehrlich down the bottom here, he was a famous microbiologist in 1913 in Germany, and he worked on fungicide resistance mostly in human systems. But his mantra was hit hard and hit early, and that timing is critical. And I think that's still valid. So 102 years later, we're still pushing exactly the same message. So first, lastly, I'd like to thank the project team, Trevor Wicks and other collaborators, all the growers and agronomists and viticulturists that sent in samples. We still may be looking for some, and of course AGWA, who've provided all the funding for this project. And so thank you very much, and over to questions. Nothing too hard, please. Might have to get back to you then.
Great, thanks for that, Suzanne. Some really interesting information there. We'll move straight through to Q&A, so please start sending your questions in. Just a reminder, if you're typing in your questions, please do so at the bottom right-hand section of your control panel. If you've got a microphone and would prefer to speak directly with our presenter, please click the raise hand button and I'll unmute your microphone.
Got resistance issues out there that you think? Oh. Is anyone, can I ask, are people concerned about resistance out there? Are you concerned in your vineyard that, well, you don't have to say about your vineyard necessarily, but you can be anonymous. But yeah, I guess that's something we really are keen to know about. If you are concerned that you think you've got resistance, then we'd really like to hear from you. Any comments there? Anyone comment about what they've seen? Nope, okay.
Yeah, no comments or questions as yet, Suzanne. We'll wait another minute or so and just see if anyone's got anything they want to add or any questions about what's been discussed today. Otherwise, we'll probably start to wrap up.
Oh, sorry. Okay, we've got one question's come through. I'll read out the question. How do you determine when analysing in the lab the percentage of isolates that are resistant? i.e., which diseases can you sample with single spore analysis? And that was from Dave Antrobus. Okay, the percentage of isolates, yes, so single sporing. Okay, so what happens is that I'll perhaps explain to the rest of you what we do. So the sample will come in, and for scientific rigour and really enable to be published and get peer-reviewed publication, we have to do what we call single sporing. So it means we take one single spore from that entire sample and put it on a leaf and grow the culture from there. So all our testing is done on what we call single spore isolates. So in that population, it's a numbers game. So if that population has a high level of resistant isolates, a high level of resistant cells in it, we are more likely that we're going to, just by chance, by picking that, we take a single spore out of it, it's likely to be resistant. Or it's likely to have that be the resistance if it's in a high level in that population. Did that make sense? If we were unlucky, I guess, and we picked out a sensitive individual, then yes, that sample could come back as testing for reduced sensitivity, and it gives us a false picture. So in something like Botrytis, you can test many single spore isolates. You can have a sample come in, and they can, let's say, 20, you can take 20 single spore isolates from throughout that sample and test them. Now with powdery and downy, it's pretty much impossible to do unless you have maybe 3 or 4 extra technicians to help. Ideally, that would be better, because then you do get a picture of what's in that population. You get, yes, because it could be that I pick out that, I just happen to be lucky and pick out that resistant cell out of that whole population. So that's a good question. It's one that I've struggled with, and because testing in a bulk population, like everything, whole populations, gives a more accurate picture of what's in the vineyard. However, it's very difficult to analyse genetically, and it's not considered sort of very easily peer-reviewed in terms of our writing papers and that sort of stuff. So it's an interesting question. I don't know that I can answered it well enough for you. I'm not sure, but that's really, and you've asked, okay, sorry, which diseases we can do everything with single spore except for downy. Downy is difficult because the spores swim around and it's generally not done single spore, but powdery is all single spored, and Botrytis is done with single spore. Most pathology work will be done with single spore analysis, but it still could be that you don't get exactly the right picture because by chance you've picked the resistant or the sensitive one that's sitting by itself. But generally it's a numbers game. If you've got lots of sensitive individuals, you're more likely to pick it. Okay, I've got another question. Is resistance to 2 or more groups at once common? From Ben Johnston. Common. I think to 1 or more groups, yeah. Probably, how do I answer that? I think it's probably becoming more common. I might defer that actually. I don't know that common's the right word to use. Certainly, if you're using those fungicide groups, then yes, it's going to become more common. Probably true resistance is, well, I guess if we look at the Botrytis data, this is the first time we've found resistance to 3 or 4 groups. So, and they're not very common if you look at the numbers throughout that whole, throughout all the samples we've got. So let's say not common, but it's potential, yeah, as we apply more fungicides and from different groups. Oh, okay, Dave Antrobus has replied, great that you are doing it that way about the single sporing. Thank you. Yeah, it was tricky, Dave, in the beginning, because really sort of industry go, well, why just testing 1 spore? But it sort of has to be done that way. So, and as I said, it's a numbers game. But really, we are getting, certainly for the QOIs, we are getting what I find in the lab and what the genetics are showing us are matching up reasonably well, apart from we're getting this mixed population. It's called heteroplasmy, and it's meaning that it's where the target site of the fungicide is. It's actually in the mitochondria in the cell, and therefore it's a different type of genetics than if it was in the nucleus of the cell. So those of you that can think back to your biology days, then yeah, it changes where the target site of the fungicide is in the cell, also changes the way resistance is expressed. So yeah.
Thanks for going through those questions, Suzanne. That's all we've got at this stage. Did you want to make any final comments, or did you have a take-home message before we start to wrap up?
I think the take-home message is that, yeah, that it's to me, and I'm new to this area, I've been working on resistance for only 2 years, so I'm not Trevor Wicks or Barbara Hall, who's been in this area for a long time, but to me, this fungicide resistance is not going to go away. It's not going to, you know, it's a problem we're going to have to deal with, and it's only going to get, I think, worse. And so keeping the chemistry that we have and being careful with it and really following the guidelines is critical, because we are going to, these chemistries are not becoming easy to come by, are they, new chemistries? So it's really critical we keep what we've got and be mindful of how we're applying them and when we're applying them. That's probably what I'd like to say. Thanks.
Okay, thanks for your final comments there, Suzanne. And a big thank you for coming in and agreeing to present today and filling in for Barbara. Thank you also to all our attendees who took part in today's session. You'll receive a follow-up email with a link to the recording of this session and also a link to a survey. Should you need to follow up on anything, please feel free to contact the AWRI directly, and if need be, we can always forward your inquiry through to Suzanne. Next week's webinar is presented by Paul Smith from the AWRI, and the topic is what are the relationships between grape chemical composition, grape allocation grade, and final wine style. If you haven't already registered for this session and would like to do so, please visit the AWRI website. And finally, thank you again to everyone who's joined in today, and please check out the AWRI's webinar program to register for future sessions.